muscle physiology audio lecture. So the first part of this one, we looked at all the um, structural stuff that makes up our muscles, and now we're going to take all that structural stuff that we learned from the first part of this chapter, and we're going to see how that contributes to creating these contractions. Okay, and then again, contraction is we're looking to kind of shorten that muscle and sort of produce tension or produce force with that muscle. But this is kind of like our first chapter where we get into some real physiology and we get into some real a little more complicated kind of steps. So you want to just kind of work through these piece by piece, and we're going to go over everything kind of a bunch of times, and we're going to be a little bit more repetitive, but it's just going to be able to kind of help us kind of remember sort of what's going on. And again, you want to make sure that you can put these contractions and kind of know exactly what's happening in each step, and make sure that you know how each step kind of leads to the next step. Okay, and we want to really kind of coordinate all those structural things that we learned in the last part and know what their function is in contributing to these steps, okay, and in contributing to creating this contraction. Okay, so the first thing we want to look at is just like a little overview. And again, when you're talking about a muscular contraction, you're really talking about this sliding filament model, which again, you guys might have heard about in high school. Okay, but everything, this is like the three, if you want to sum it up into just three basic steps, everything is going to begin with this stimulation or this impulse from our nervous system. Okay, so it begins when our nervous system sends an impulse to our muscle fibers. Okay, and then what we're looking to do is with this impulse, we're going to create these cross bridges between our thick and our thin filaments, which is what we talked about last time. And then once we form these cross bridges, the sliding is going to begin, and this is really going to be, when you break it down to just the simplest sort of explanation, is these thin filaments are going to get pulled towards the center of the sarcomere, and that's going to be by the globular heads on the thick filaments. Okay, so this is the three basic things that are going to happen when we're contracting a muscle, and then now we want to dive in and sort of look at each step and sort of how each one of these things happens a little more specifically. Okay, and we want to come back to this picture, and again, this one is a great one to kind of have beside you as you're working through the chapters, and you can kind of, we're going to talk about sort of what changes when a muscle contracts with each of these little segments here. But it's good to have these little images, because then again, I can read over the explanation, and I can go back and I can find those structures, and I can look at the A band and the I band and the Z disc, and I can imagine a little bit better what's exactly happening when we form these cross bridges and we have this contraction. Okay, so a good one to kind of go back and just review and sort of have next to me as we work through all the steps. Okay, so during a muscular contraction, and again, this is a really good multiple choice question, because they can be like, which one of these does not happen when a muscle contracts, or which of these is false, or which of these is true. But these are all the things that are going to happen and kind of change with these pictures here. This is a list of all the things that are going to kind of change when you contract a muscle. Okay, and we're going to kind of list them, and then we're going to go back and kind of look at each one a little more individually. So the thin filaments are going to slide centrally, and again, we always want to remember that the thin filaments are the ones that are going to move. Okay, they are the ones that are going to be pulled centrally. The thick filaments are going to stay put, and the distance and the size of them are not really going to change. It's just going to be the heads that are going to kind of pull on things. The Z discs are going to be pulled towards the M line. Okay, so we're going to go back and take a little picture and make sure we know what that M line is, and make sure that we know these Z discs are going to get pulled again towards that M line. The I bands are going to shorten. Okay, the distance between successive Z discs is going to shorten. The H zone is going to disappear, and then the A bands are going to move closer together, but their lengths again are not going to change. Okay, so it sounds like a lot, but again, when we take the picture and we sort of list all these things, we can kind of piece it together a little bit easier. Okay, so the thin filaments are going to slide centrally, and again, these little blue thin actin filaments are the ones that are going to slide, and the ones that are going to be pulled by these myosin filaments. So these little filaments are going to get pulled in this way towards this central line here. Okay, the Z discs are going to be pulled towards the M line. Okay, and again, the Z discs are kind of like the line here that are attaching these, they're attached to these kind of actin filaments. So as these actin filaments get pulled centrally, it makes sense that these little Z lines here, Z discs, are also going to get pulled again towards this sort of midline. And then the distance between this line and this line, as they get pulled centrally, is obviously going to be a little bit shorter. Okay, so the distance between successive Z discs is going to shorten. The I band is going to shorten, so we want to look at this I band, and we want to know that, okay, the I band is the distance between the ends of the myosin filaments here and the myosin filaments on this side. And as I pull these kind of centrally, as they attach to the actin and the actin gets pulled towards the midline, these little ends of the myosin filaments are going to end up being kind of closer together. And we're going to kind of see a picture of that too here in a second here. The H zone is going to disappear. And again, the H zone always gives people a little bit of trouble because it kind of looks like it's these red parts of this that they're talking about, but it's actually the distance between the ends of the actin here. It's that little space between those two little ends of the actin that's that H zone. And again, as these actin filaments get pulled centrally, this little H zone is going to totally disappear, and those actin filaments are going to be pulled towards the center. The A band moves closer together, but the length does not change. And the A band is the length of the myosin filaments. Okay, and again, this A band is going to get pulled closer to this A band, but the actual length of those myosin filaments is going to stay exactly the same. Okay, so really, really important to remember that it's the actin that moves, it's the actin and the Z discs that are pulled. The A band is going to do the pulling, but the size of it is going to kind of stay the same. And here's a picture of some actual images of the muscle fibers. Okay, and I can see with this one, this is where my muscles are relaxed, and this is where my muscles are contracted. Okay, so you can see in this one, I can look at the ends of the actin. I can look at this one, and again, they've moved towards the midline. Okay, so that's going to be that H zone here. I get my Z discs up here, and again, they're going to move a little bit more centrally. And I can go through my list again, okay, and I can just make sure that I kind of understand from this picture to this picture kind of what's changing and why it's changing. Okay, one of the best things that you guys can do is kind of make sure that you can draw this. Okay, so now you don't have to be a really good artist, but you just want to take a blank piece of paper and sort of draw this out and sort of put the lines in and make sure you can label everything perfectly and get the A band and the I band and the sarcomere and all those things. And if you could just sketch it, even if it's really, really messy, if you can make sure that you can kind of sketch it on a piece of paper, then you know it. And then when I go back and I'm trying to kind of figure things out, I can kind of go by that drawing and I can draw it out and I can figure out exactly what's kind of happening. Okay, but this is all the things that are going to happen when we contract those muscles and we're attaching those filaments and we're kind of shortening this muscle and producing all this force. Okay, so now we have to go back to the beginning, and now we know what kind of happens structurally and what changes with all these different little segments. But we want to go back to the beginning, and we want to kind of work it kind of step by step from that first electrical impulse, okay, to where we're going to have that shortening and we're going to have these contractions. So we go back to the beginning and we say, for a muscle to contract, our nerves must send an impulse to our muscle. So again, it all starts with this impulse from your brain, your spinal cord, from your nervous system, okay? And this impulse is going to reach the muscle at the neuromuscular junction. And this is, again, where we start throwing in these big terms, but the name kind of tells you exactly what's going on, right? And you don't want to make it really complicated. You just want to be like, okay, neuromuscular junction, it's where the nerve is going to meet the muscle. And that's exactly what's kind of happening. And if you look at this picture, I have my muscle here, and I have my sarcolemma, and I can see my nerve, and I can see where the nerve is kind of coming into contact with that muscle. And this is going to be that neuromuscular junction. And then we're going to talk about exactly kind of what happens when that nerve kind of gets to the end, to its end, and how it kind of interacts with the muscle. Okay, so now we have somatic motor neurons, and again, these are, soma means body. So it's going to be nerve cells that activate skeletal muscle fibers. Okay, they're going to reside in the brain and the spinal cord. They're going to have extensions. Okay, so coming off of my spine, I have these extensions on my spinal cord. And again, they're going to be my nerves, and they're going to be called axons, and they're going to extend into my muscles. Okay, and these axons are going to branch out as they enter the muscle, and they're going to form these little junctions with the muscle, and that again is going to be called this neuromuscular junction. Okay, at the ends of the nerves, I have these axon terminals. Okay, and that's going to be the ending, and we're going to see a picture here in a second. But they're going to have all these little it's almost going to be a little bulb, and they're going to have these synaptic vesicles at the end, and they're going to contain these neurotransmitters. Okay, and the one that's important for these somatic motor neurons is going to be this ACH, or this acetylcholine, and it's going to be part of the step that we're going to walk through as we get to creating these contractions. So now we come back to this picture, and again, we have our axon, we have our terminal, right? And then we want to remember that that end, this little bulb at the end here, is going to contain all these little neurotransmitters. Okay, so here's a picture of the muscular part. I have my nerve, I have all my little branches, and I just want to remember that at these little terminals, these are going to be filled up with all these little neurotransmitters that I can kind of stimulate, and what's going to happen is I'm going to release all those neurotransmitters. Okay, so these are the events at the neuromuscular junction. Okay, and number 1 is an action potential. And anytime you see action potential, we're going to come back to those a lot when we get into the neurological chapter, but action potential just means impulse. And the reason it's a little tricky is that it can be named a bunch of different things. But anytime you hear electrical impulse, action potential, they're basically talking about the same thing. Okay, but this electrical impulse is going to arrive at the axon terminal, so the end of the nerve where all those neurotransmitters are. Okay, and what's going to happen is that as that nerve gets towards that terminal, these voltage-gated calcium channels are going to open. So the cause of that transmission of that electrical impulse is that we're going to open up these calcium channels, okay? And calcium is going to enter the axon terminal following the concentration gradient, not super important, but the reason or what's going to happen when we open up these calcium channels and that calcium flows in is the release of that neurotransmitter, okay? So the calcium entry causes the acetylcholine neurotransmitter to be released. It's going to diffuse across the synaptic cleft. We're going to see that in a picture here in a second. And it's going to bind to the receptors on the sarcolemma. And again, you want to think of that sarcolemma as the plasma membrane of the muscle cell, okay? So you can come back to this picture, and I'm going to show you another picture in a minute where you can kind of do all the steps, and you can make sure that with the picture you can follow through and sort of know exactly what's going on. But it's nerve impulse, right? Gets to the ends where those terminals are. Again, these little terminals are filled up with neurotransmitters, acetylcholine. When that nerve impulse gets to the end of the nerve, it's going to open up these little calcium channels here. Okay, and that calcium flowing in there is going to cause this nerve or this synaptic kind of bulb here to release its neurotransmitter or its acetylcholine. Okay, and then what happens with that acetylcholine is it's going to bind to these receptors on that sarcolemma. Okay, so number 5 is the binding opens ion channels in the receptors. So once those channels kind of open, okay, it's going to allow passage of sodium into the muscle fiber. Potassium is going to move out of the muscle fiber. We're going to have more movement of one than the other. Again, not super important at this particular point, but it's going to cause this little action potential to kind of run along that whole sarcolemma. Okay, so what's going to happen is we have this little release of these neurotransmitters at one segment, and what that's going to do is it's going to cause this electrical impulse to kind of flow along the whole sarcolemma. Okay, and then we know the sarcolemma is kind of linked up with that T-tubule. We know it's kind of linked up with the sarcoplasmic reticulum, and that's how we're going to get in towards those little actin and those myosin kind of filaments. Okay, so here is a great little photo. And again, I can kind of work through all the steps, right? So I can work through the step. I can be electrical impulse coming down my nerve, right? It gets to the end here, and we want to be like, okay, so what happens when that electrical impulse gets to the end? It's going to open up these little calcium channels. The calcium is going to flow in there. What's that going to do? It's going to cause this axon terminal to release its acetylcholine. The acetylcholine is going to bind with the receptors on the sarcolemma, and then that's going to cause another electrical impulse to flow along this whole sarcolemma, okay? And then we're going to move on to kind of the next part. But you want to kind of work through each of these, and each time you add a step, go back, do the steps again, and then just add the next step so that until we get to the end. Okay? But this is a great picture to just be able to like, okay, what's going on in each step, and how does each step sort of move on to the next one, okay? And this is number 5, and this number 5 is really just how I'm transmitting this electrical impulse along that sarcolemma, and it's going to come down to these little sodium-potassium kind of channels. And we're going to come back to the sodium-potassium channels when we get into the neurological chapter. Okay, I also have these little videos in here. I'm going to let you kind of watch these on your own. But these are just some great videos that talk about all the things that we just talked about, but it just lets you kind of hear it from another perspective. And these are great to sort of, learn all your stuff, learn all your steps, and then go back and watch the little videos and make sure everything that they're talking about and all the steps that they're talking about kind of makes sense. And if you can kind of, follow along and everything makes sense when you're watching the video, then you know that you're ready to go. So now we get to how do we turn this release of the acetylcholine and the production of this action potential along that sarcolemma, how do we turn that into a muscle contraction? Okay, and the proper term for that is this excitation-contraction coupling. So we get another big kind of tricky little term here, but basically what that means is that it's the sequence of events by which the transmission of that action potential, okay, or that electrical impulse, along the sarcolemma is going to cause those myofilaments to kind of form these cross bridges, and it's going to cause them to slide, okay? So we're back to our sliding filament kind of model here. And we want to go back to this one, and the reason this one was so important in the other one is that here is that sarcolemma, and again, at this point you have this electrical impulse traveling down the sarcolemma, say, or along the sarcolemma. And remember, this sarcolemma is hooked up with this little T-tubule here, right? So the T-tubule is kind of a part of the sarcolemma, but this is how I'm going to get this impulse sort of transmitted into all these muscle fibers and into this little sarcoplasmic reticulum. Okay, so the electrical impulse goes down the sarcolemma, it gets transmitted into my little T-tubule here, okay, and then it can send that impulse to the sarcoplasmic reticulum, and then I can move into all of the next steps. So again, really, really important just to remember that this T-tubule is connected to this sarcolemma, and again, it's going to be the part that allows me to transmit that impulse into all these little muscle fibers. Okay, so what's happened so far, and again, we're going to go back and kind of go over the steps each time we sort of add a step, just because the repetition will kind of help us to remember. So we had a nerve impulse that reaches the axon terminal. What's going to happen when it reaches the axon terminal? It's going to open calcium channels, okay, and that calcium entry is going to trigger the release of that ACH or that acetylcholine, okay. That ACH is going to bind to receptors in the sarcolemma. It's going to open up those sodium-potassium channels, and it's going to create this action potential that's going to spread across the whole sarcolemma, okay? And it's going to transmit that action potential along those T-tubules, and that's going to stimulate my sarcoplasmic reticulum to release calcium. Okay, so this is, again, the first kind of tricky little physiological stuff that we have to sort of learn, but it's nerve impulse, okay? Release of acetylcholine, and then we say, what does that release of the acetylcholine does? It opens up these little channels here, okay? It's going to create this action potential that's going to spread across that sarcolemma, and then what's going to happen when that action potential spreads across that sarcolemma? It's going to go down this T-tubule, and it's going to stimulate that sarcoplasmic reticulum to release my calcium. Okay, so this is, again, an important page just to kind of work through each of the steps, right? Nerve impulse, open my calcium channels, release my acetylcholine, right? Spread this action potential along that sarcolemma, down the T-tubule, into that sarcoplasmic reticulum, which again stores calcium and is going to be the part that's going to release that calcium for me. Okay, so get this part down, know all the little steps, and then we're just going to add on the last part, which again is the cross bridges and the sliding. Okay, and then here's a picture of a close-up of, here's my sarcolemma. So again, that impulse is coming along the sarcolemma. It's going to continue down the T-tubule, and this is where it can stimulate that sarcoplasmic reticulum to release its calcium. And you can see all the calcium kind of stored up in that sarcoplasmic reticulum, and I can also see when I release that calcium, I'm coming into contact with all these little actin and these myosin filaments. So good to kind of know the steps, but again, the pictures make it really, really good to kind of make sure that you can follow along. So now we get to the cross-bridge part, okay, and we have these cross-bridge recycling or the muscle fiber contraction. So cross-bridge formation is going to require calcium. As calcium levels rise due to, again, that nerve impulse, they're going to bind to troponin. So again, we talked about the actin, and we talked about troponin and tropomyosin, and this is where that calcium becomes really important. And remember, on that actin, we had those binding sites, but they're blocked by this tropomyosin. Okay, so that release of calcium from that sarcoplasmic reticulum is going to bind troponin, okay, and then this binding of calcium is going to cause a shift in the tropomyosin, because we want to remember the troponin was the binding site, the tropomyosin was the part that was blocking the binding sites, okay, so this release of calcium binds troponin, causes a shift in the tropomyosin, and this unblocks all the binding sites on those actin filaments. Okay, so really, really important to sort of remember, like, why is that release of calcium important from the sarcoplasmic reticulum? This is why it's important, because it's the part that's going to unblock those binding sites for those myosin heads. Okay, so here is our little picture here again of our little actin, and I can see where those binding sites are blocked by the tropomyosin, and then we just get a little reminder of, okay, what did each do? So the tropomyosin blocks the sites, the troponin again is that binding protein for that calcium. So therefore, when calcium is present in sufficient quantities, okay, the myosin binding sites are unblocked, and then this cross-bridge cycle can occur, okay? And the cycle is going to repeat as long as there is adequate calcium and ATP present to be able to use. So as long as I can keep those binding sites unblocked and I have the energy to sort of keep moving these contractions along, I can sort of, continue this contraction as long as these two things are still present, okay? So with each step, the myosin head is going to attach to the actin further along, and we're going to look at a picture of that in a second here too, okay? So continued nerve impulses equal more calcium. Contracted muscles can shorten by 30 to 35%, which is an enormous amount. You can just see here if you straighten your arm and you look at how long your bicep is, and then you bend your elbow and you look at how short your bicep gets. It's a pretty significant amount. Okay, so each myosin cross bridge attaches and detaches many times during a single muscular contraction. This question always kind of comes up, and people ask, like, okay, well, when they attach and detach, why does the muscle not slide backwards? And when you look at that little picture, you can tell that there's a lot of little myoglobin heads, right? There's a ton of heads, and at some point, some of those heads are kind of always attached, and this keeps the muscle from kind of sliding backwards until we reattach more heads. Okay, so here's a picture of our cross bridge formation. And you get the little images here that are kind of reminding you that this, our body has to use energy to kind of do this, okay? But this is the myosin head attached to the actin. And again, the myosin is going to be the part that's going to pull the actin towards that midline. And the actin is the one that's going to kind of move, okay? And then it can detach, get some more energy, reattach, and then do the same thing over and over and over as long as I have that nerve impulse, I have calcium, and I have energy to sort of do all these things here. Okay, so here are, again, just the little steps for a thing. You don't have to necessarily know the steps. You just want to remember sort of that it requires energy. It's the myosin binding to the actin. And remember that you need the impulse, you need the calcium, and I need that ATP to sort of continue my contractions. Okay, and this is another video that you can watch. Do the same thing where you kind of work through the steps on your own. Make sure you can kind of write them out, and then you can watch the little video and make sure that, again, that each little step in the video kind of makes sense, and that you're if you need to go back and sort of review anything, you can do that once you watch the video. Okay, so here's a little review. And again, we want to make sure this is the first part. We want to make sure we know all the steps to kind of create these contractions. And this is kind of the simplest way to kind of walk through it. Okay, so it had nerve impulse, reaches the axon terminal. Okay, it's going to open up my calcium channels. The opening of those calcium channels, or the entry of that calcium, is going to trigger the release of the neurotransmitter acetylcholine. Acetylcholine is going to bind to the receptors on the sarcolemma. That's going to open up all these sodium-potassium channels, and it's going to create this action potential that's going to spread along the sarcolemma. And we want to remember that the sarcolemma leads to the T-tubule, okay? So that impulse is going to spread down the T-tubule, and it's going to stimulate that sarcoplasmic reticulum to release the calcium. And now we've kind of looked at sort of why is that important, right? So as the calcium levels rise, they're going to bind to troponin. That's going to cause a shift in the tropomyosin. It's going to unblock the binding sites on the actin, allow those cross bridges to kind of form, okay? And then again, the cycle can repeat as long as you have enough calcium and you have that ATP present to sort of keep, you have enough energy and you have the binding sites unblocked. You can keep sort of repeating that cycle as long as you need to. Okay, so with each step, again, the myosin head's going to attach and detach and kind of pull that actin a little bit closer to that midline. This is a picture that I was kind of talking about before, and this is kind of a great one because I can do all the steps that we just kind of talked about, right? I can have my, I have my nerve, I have my neuromuscular junction, and I can kind of start from up here, and this is the way that I kind of like to do it, is you can kind of go, okay, here's my nerve impulse coming down, right? Reaches the terminals. It's going to open up those calcium channels. That's going to cause the terminals to release their acetylcholine. The acetylcholine is going to be released. It's going to bind with the receptors on the sarcolemma, okay? It's going to cause this action potential to be spread down the sarcolemma, down the T-tubule. It's going to stimulate my sarcoplasmic reticulum to release its calcium. The calcium is going to cause the unblocking of those binding sites, okay, on the actin. It's going to allow those cross bridges between the myoglobin and the actin to form, and then I can use my energy to sort of pull those actin, and I can create this shortening kind of cycle. Okay, so this is a great one to just sort of look at the picture. You can walk through all the steps and sort of make sure, again, that you know each of the steps, but that how you know that each of the steps kind of leads to the next step. Okay, and this is just a zoomed in picture again of the sarcolemma, the T-tubule, and again, just that stimulation of, or the transmitting of that action potential down that T-tubule stimulates that sarcoplasmic reticulum to release its calcium. The calcium unblocks the binding sites, and then I can get my cross bridge formation, and I can get the pulling of that actin towards the midline and my muscle shortening. Okay, and then here's just a couple other little pictures of just those cross bridge formations, and then just the pulling of the actin. For part 3, we're going to move into We talked about in this part, we talked about how we need energy to sort of create these contractions and continue these contractions. In part three, we're going to look at how do we provide the energy to our muscles to sort of do these muscular contractions, and we'll look at just a couple other little contraction terminologies. Okay, and then we're done. After part three, we're done for all of our material leading up to our next set of exams.